Wonderful. So thank you everybody for joining our 10th month in the CGC monthly webinar series. And this is the last webinar of this year. Any resources from this presentation, including the slide deck and the recordings, will be posted in the Cancer Genomics Cloud website. So it's cancergenomicscloud.org/webinars. I'm really happy to welcome all of you to this webinar titled HLA Class 2 across TCGA. A couple of announcements. Please use the chat box for asking questions. At the end of the presentation, we will have a Q&A session. And we will also post a link to a feedback survey at the end of the presentation. So please respond to that. And you will also get the link in your email tomorrow. So if you forget to respond to the survey today, please do that tomorrow. It will help us in improving our presentations. Like I just mentioned, this is the last webinar of 2021 because we are moving into holiday season next month, and the webinars usually happen on the fourth Wednesdays, and next month and the month after that, it's close to the holidays, so we will not be having the CGC webinar series. We'll probably reconvene again in January. You will see the announcements soon. So it is my pleasure to introduce our Collaborative Project Award recipient, Dr. Pascal Bellew, who is here with us today to share his research work performed on the Cancer Genome Atlas datasets using the 7 Bridges Cancer Genomics Cloud. Dr. Bellew has a degree in math, and he worked as a bioinformatician in Quebec City, where he gained expertise in the genetics of bipolar disorder and then worked on software and model development for gene expression response to steroids in breast and prostate cancer in mouse models. As a graduate student, he developed expertise in the genetics of modifier genes in glaucoma and in Monte Carlo Markov chain MCMC analysis. Currently, Dr. Bellew is a computational biology postdoctoral fellow in the Krasnitz lab at the Quantitative Biology Department of Cold Spring Harbor Labs. Dr. Bellew has been working on machine learning methods for prediction of drug responses based on genomic information in pancreatic cancer. And his main interest is to model genomic information related to pancreatic cancer in the objective of gaining a better understanding of the disease. Today, he's going to talk to us about the work he performed on HLA class 2 typing across the entire TCGA collection of approximately over 11,000 patients spanning 33 tumor types. So without much further ado, over to you, Dr. Bellew.
Okay, thank you. Yes, I thank the 7 Bridge to give me the possibility to present today my work on HLA. The work is not complete yet, but we progress, and probably we will finish relatively soon. So I will talk about HLA class 2 and the cancer sample from TCGA. The objective is first characterize the HLA class 2 and all the samples from TCGA. This will give us the possibility to identify some target for immunotherapy and other information. It could be too interesting to see if they have link between HLA, and some could be used as a marker for cancer, for the progression of the cancer. Sorry. So HLA are gene with triggering the immune response. You have the principally 2 class of HLA gene, the class 1, which they are on the first part of the chromosome 6. And you have the class 2, which one we will talk about. They trigger the immune response via the CD4 T cell, and yes, and these genes are really polymorph, and so they are a little bit hard to characterize because the problem of mapping and etc. So we have some specialized tools to work with the HLG. And another aspect, they have a specific way, a specific nomenclature to typing each gene. So you have example here, the nomenclature for HLA gene A. This is a class 1. So you have the first one are the name of the gene. After you have the third, okay, we call field or digit. They are a number characterize a cluster of binding, potentially binding cluster of the gene. And the second field corresponds to identify a specific protein. So if they have some change or variant in the gene that change the protein, the number would be changed. The third one are the synonymous variant, and finally they have the non-coding number for the number to characterize the non-coding number, the non-coding variant in the gene. So for each gene, you have a big database that identifies a number with a specific haplotype, so a specific sequence for the gene. For the typing, we use 7-bridge to run on all the TCGA sample. We have two principal tools, XHLA. We use XHLA on the normal exome to type each sample, and ARCAS HLA for the RNA-seq. We did a lot of tests with the other tool and HLA profiler in particular. It was pretty good, but we finally opt for ARCAS HLA because it's a lot faster, so they reduce a lot the cost of the processing. I work a lot with the expedition. All the results are shared with expedition working with us at Cospring Harbor, particularly with Dave Twenson. And yes, so we use XHLA to type each patient from the normal exome, and ARCAS HLA, we type the same patient, the majority of the case, but using the RNA-seq. In fact, we have run XHLA on 9,021 exome from normal tissue, so the majority are blood, and some solid tissue, but the vast majority are blood. Some of the exome are from the same patient, so we have 8,900 case, so more patient run. With XHLA, we type 3 genes from the class 1, gene A, B, C, and 3 gene 2 from the class 2, DBP1, DQP1, and DRB1. And they don't characterize, often for the class 2, we have the alpha alpha branch and the beta branch. So, but XHLA don't characterize the alpha. For ARCAS, we run for the RNA-seq. They are tumor sample. We run it on 10,000 sample from near 8,000 cases. Again, the gene type from the RNA-seq are the class 1 ABC. We have a lot more class 2, DMA, DMB, DOA, DOB, DPA 1, DPP 2, 1, DQA, DQB, DA, and the different beta branch. And some non-classic gene, actually class 1 non-classic, EFG. They have 6,000 sample in common, case in common. They are not necessary. They are not sample because the XHLA run on the normal tissue and ARCAS run on the RNA-seq tumor tissue. So we have 6,000 in common. For validation purpose, we use 5,400 validation. We will discuss about it from the primary tumor. In the case of ARCAS, we don't use the metastasis and other type of sample. So if we compare the results from the results from the RNA-seq versus the normal tissue using XHLA, they have relatively a lot of validation that the results are relatively are a lot better than this to the correspondence between the XHLA and ARCAS or the other two. But we have to keep in mind that XHLA run on the normal exome and ARCAS run on the RNA-seq cancer. So they have a lot of change because of the expression and change because of the cancer. So if we look at the class 1, we validate around 75% of the gene for the gene class for the gene ABC. So in red are the alleles that don't correspond to the between XHLA and ARCAS result, which is a pretty big difference. But we have to study a little bit more to explain what is exactly the difference. You have probably a difference because they're RNA-seq, and we want to rerun the same analysis on the cancer exome to see the contribution of the change in the exome in the cancer's gene from the HLG gene in the cancer. We have changed that explained a part of the error of the difference, and the expression play probably a pretty good difference too. If we look at the class 2, again, it's a little bit, in fact, they are a little bit worse. They are they valid less, and this is a little bit no. It's they have more difference in the class 2 between two and between the RNA-seq and normal and the DNA. and the cancer and the normal. So we are a little bit near more 60%, except the DRB1. We have a lot, it's a lot better, but they don't have the diversity of the typing are lower for DRB1. So and the comparison is just made on the first digit. And yes, the diversity is lower, so it's easier to get a better correspondence between the two analyses. So if we look at the TCGA allele, so we take the gene A, we have the first digit, like I said, is the cluster of second binding by the gene. So we have the digit 02, 24, 1, 26, and 68. And this is the typing based on with the ARCAL. The first line is the frequency of this allele in the TCGA sample. In TCGA without the replicate, so I remove all the samples from the same patient. So for the 0.2, the digit 2, we have 34% of the alleles in TCGA. 24 is 12%. 01 is 11%. And I guess from allele frequency, they compile a lot of studies that characterize the HLA gene, often for immune questions for transplantation and stuff like this. So they have big samples where they characterize the allele. So if we look at the sample, I select few samples. I select often TCGA, it's a majority of Caucasian people. So I took big sample, more than 1,000 from Caucasian groups. And we get something relatively near the result for the digit 2. Maybe Italian one is a little bit lower. Same thing for the 24, and maybe the 26 is a little bit different, but again, they have some variability, and they characterize less not all the studies characterize detected. If we do the same thing with one gene, I did it for all the genes, but it will be a little bit. So I use DPA and DPB, the alpha and the beta, in the same gene, not two genes, but with alpha and beta. So in this case, the vast majority of DPA are from the digit 1. And we saw the same thing in the Norway sample and T sample. You have a lot less study for the HLA class 2. So it's the reason why I didn't have the big study like Italy and the one sample, 100,000 sample. If we look at the BP2, this one has some difference. In TCGA, we have 0.25 for the digit 1, and all the Caucasian samples are a lot less in the population. We have something similar too for the digit 3. And finally, we have the digit 4 that are a lot more represent in the population sample and less represent in the TCGA. So at this point, we look to if we have some characteristic more specific to some cancer or tissue, because of course the data are from RNA-seq. So the expression could be modulated by the tissue. So if we change cancer, we have a covariable that it changes the tissue often too. So I use the primary tumor. So we restrict the study to the primary tumor in this case, and we look at the list of the primary tumor. We have in TCGA in the sample we have typing, and we have 26 primary sites, and we keep 18 primary sites where the sample have more than 100 sessions for each site. So in total, we use 6,800 sample case. In fact, 7,000 if we include the small tissue. The tissue with more than 100 are generic brain, the brain, the colon, colorectal, esophagus, head, neck, kidney, liver, ovary, pancreas, prostate, skin, soft tissue, stomach and testes, thymus and thyroid. We have some technical issue with the breast. Probably we will integrate later. Yes. So this is the distribution of the sample. We have the list of the different tissue corresponding probably to the different cancer. And we have the number for each one, for the 6,000 sample. So we have a lot more. The big one are brain. The colon is probably relatively big. The ovarian are big. Liver, colorectal. If we look at the first digit for the class 1, for the class 2, we will use again as example the gene DPPA. DPPA1. If we look from all the different primary tissue, we have the frequency of the digits. The digits are here and here. So we have digit 1 from the different one. They maintain all around 0.75. And for the digit 2, they are all around 0.25 for the different tissues. So they don't have, for the alpha branch, they don't have a lot of change. And if we look at the same thing in the DPB1, in this case, we have like a little bit in the population, we have some big difference. So first, the 0.4 is a lot more present in colon, which corresponds more, in fact, what we saw in the population. If we go back at this one, the other 4 are around 0.5 in the population, which is correspond a little bit as what we saw in the colon. But they are really low for the auto sample. they are not there because they are lower in the list. And the big, more represented for the neck is the digit 1 and 3. And for the same thing for the thyroid and the brain and the ovary have less, a little bit less. So they have some, in the case of DPPB1, they have some link between the type and the cancer. And we have some association. So we don't know exactly if this association is came from the expression because of the tissue, or they are come from the some characteristic of the cancer. This is what we have to work on it. So in conclusion, we have some interesting patterns. I don't include the clustering. We have some clustering, but we saw it's not at this step very clear what we but we have pattern a little bit like we saw in DPP1 related to the class 2 to the cancer. So we need to characterize a little bit more related to the expression. If we integrate the information, if which genes are more expressed or less expressed, so they can change our ability to type correctly or type the gene, which can create change for each tissue. And we have to check too for the cancer exome and see more specifically the difference between the normal and the cancer relatively to the cancer, relatively to the sequence. So if we work with the DNA, we remove all the information from the expression. So it will be a lot more easier to characterize. We are working the problem with to characterize the exome. The cancer exome, it's a little bit need relatively a lot of computational. So we work to optimize the process, and from this we will work more on the characterization of the HLA class 2 in the different cancers and see if you have some allelic specific expression related to the cancer, if you have some other pattern. We probably will not go a lot in the binding. It's a really interesting point, but probably we will not have time in short term to go in this. So that's it. So I would like to acknowledge our my PI, Dr. Alexandra Smith, and Dr. Dave Stevenson, and in particular Astrid Deschamps work a lot on the project. Thank you.
Great. Thank you so much, Dr. Bello. It was a very interesting presentation. There are a few questions that I wanted to ask you, and also wanted to mention to the audience, if you have any questions, please post in the chat box and we will address them now in the Q&A session. So one thing I wanted to ask was, what do you think is the application of HLA typing? I know it has a lot of impact in immunotherapy for cancer, but are there other applications that you are envisioning, especially the work that you have done on the TCGA data sets? What would be the impact of this work?
Okay. Of course, the immunotherapy could be one of the biggest inputs. We can think about if we have some pattern more specific to the cancer, so they can be used as marker for severity or for some characteristic of the cancer. So, and of course, the big point is if the susceptibility to the response to the immunotherapy will be the big point. And yes, and
Okay.
yes, this is the main application potential.
Great, thank you. So I know there are some bioinformatics challenges, right, in HLA typing. So can you talk to us a little bit about, what are the challenges that you faced, and potentially how did the cloud workspace help you in addressing those challenges?
Yes, of course, the cloud, we cannot do this without the cloud. With this, just to process the 10,000 sample, we need so we need to the big part for all the tools is we have to remap, extract. We extract the more specific sequence based on the first mapping from the TCGA data, and after we remap the subset of read, and from this subset of read remap, we look at the database. So we have different potential way to look at it. We can construct a network, a graph that will with the different variant to characterize each HLA haplotype, and after when we identify which haplotype is the most probable, and of course we look at the haplotype. So we have the phase challenge too. So we have to separate the two chromosomes if the different variants are from the same parent or are from different parents. So we have to rephase each variant to be able to characterize the haplotype.
Got it. So to summarize, it's a multi-step process, and also probably because of the sheer size of the samples, it's computationally intensive as well.
Yes, yes, and the size, yes, of course. Yeah.
Great. So we have a question in the chat. Very interesting talk, absolutely. So what is the concordance of high resolution genotyping of HLA class 2 non-classical HLA compared to inferring HLA typing based upon RNA-seq? So there's one more question, so I thought I'll give you some time to respond to that.
For the HLA class 2, yes, relatively. The non-classical, we don't have comparison. So we have only RNA-seq from ARCAS data. So we don't have any information to do the comparison. Probably when we will reprocess, it will be interesting to have this information from the exome and see if we are able to characterize this one. Yes. And
okay, yeah. There's another question. When you say failed in the comparison of normal versus tumor, did you do experimental validation, or did you just compare if normal and tumor have the same type? And
okay, yes, we just work bioinformatically. So we don't have any validation, experimental validation based on the, and yes, fail is maybe a little bit big word. It's not non-correspondence between the allele in the same patient. Uh,
Okay, so there's another question. What is the reason to select HLA and ARCUS HLA? Did you try polysolver for benchmark comparison?
No, we don't use polysolver. We use HLA, HLA-HD. We use HLA profiler. And I forget another one. And it's not Kurami. We use Kurami too. Kurami is pretty good for the whole genome. It performs a little bit less for the exome and the RNA-seq. XHLA performs pretty well for the exome. The majority HLA profiler is very good. The only probably problem is just the time to process. So ARCAS for the RNA-seq are a lot more efficient to process, but yes, and all the tools are relatively concordant. If we take the same thing, we compare the same thing with the same thing, the concordance was pretty high. And I think I can send a couple of papers to the benchmark, and they are all relatively good. XHLA are nice for the exome and the ARCAS, but it's not the only one choice, possible choice.
Got it. Thank you. So a related question is, so how do you compare the accuracy of these tools? Are you, testing them with some public benchmarking data sets, or how do you?
Yes, at the beginning, we did with data set. We have gold standard CAL for some 1,000 genome sample, and we process with all the tools the same data set and see the correspondence. And the correspondence are really good in controlled environment. They are really good. Here we search for problem because we compare normal with cancer and exome DNA versus RNA-seq. So we create a lot of problem. But on the other side, they are interesting problem because they show that they show the difference between because the expression, because the variability of the tumor create some difference, interesting difference. The only problem we are working with here is exactly characterize what exactly caused the change. So if there are some error or some more specificity to the expression related to the cancer, related to the tissue, related to the difference between DNA and RNA.
Great. We have one more question in the chat. So in the context of immunotherapy, are you looking for HLA types or supertypes that might be associated to specific cancer types? If you find supertypes, is it possible that this leads to a non-personalized cancer vaccine that can be used for a wide range of human populations?
Yes. If we find a really specific supertype, it could be a nice way to generate a vaccine for a big group. The other way is, of course, to it more personalized medicine. So look if you have some antigen more specific and in the tumor and try to target it when they are there. So yes. So you have both. We can look at the super type or less more precisely at the protein. Yeah.
Okay. One more question I have is, so you briefly spoke about the next steps about using the exomes in your results and also the expression data. So what are the, like concrete next steps? Do you need to, develop some pipelines? Like what kinds of tools you will be using?
Yes. Probably we will have to, for the integration, we have to reduce a little bit the cost for the cancer exome. So it's one part. It's an optimization of the process. We need to some tool for the integration. So we are working on it for integration of all the information together and create a way to characterize all that part of the data set. Yes, of course, we target to use, majority is probably use the existing tool, just more try to get the most efficient way to process, and some in-house tools for the integration. A little bit for the HLA. We have some hypotheses we work on some dataset in the yes, and maybe I know you have the graph tool from the this we have to work a little bit more, but if we are able to do something really efficient in a graph way, it will be really interesting.
Great. If there are other questions, please post in the chat. I'll give another few more seconds for everybody to post their questions. Yeah. So that was very, very interesting. Thank you so much for your presentation, Dr. Bellew.
Thank you.
We were very excited to hear, and we would like to hear more about, how this progresses in the future.
Yeah.
All right. I would like to thank everyone for joining us today for this presentation. And just a quick reminder that this is the last presentation for this year. We will not be having CGC monthly webinar in the next 2 months. Please note that and enjoy your holidays in the next 2 months. With that, I would like to conclude today's webinar. And thanks again, Dr. Bellew, for your talk. Bye, everyone.
Thank you. Bye.